Forensics means connected with the courtroom. Thus, forensic science is concerned with gathering hard evidence to be presented at trial. The foundational principle for forensic analysis is that every contact leaves a trace, with genetic markers, also known as gene loci, generally having strong variation which can be observed and recorded.
Short tender repeats is a major genetic marker used in forensic study. It consists by 2 to 6 base pair and repeats thousands of times. It can be examined by PCR, and as the number of STR repeats are highly variable between two people, it identifies as a unique signature for individual.
Polymerase chain reaction, or PCR, takes a tiny amount of DNA that was left at a crime scene and turns it into a sample big enough for testing. It does this by heating and cooling the DNA within a pool of nucleotides, which are DNA's building blocks, to help it replicate into many identical copies based on the first one. This new amplified sample can then be digested by restriction enzymes, which snip out the STRs and run them through an electrified gel in a process called gel electrophoresis. This test separates them by size, showing us the genetic fingerprint of that individual.
As we all know, DNA profiles are highly symbolic to an individual, but how could they help to solve a crime? Police collect the DNA sample from a wide range of convicted offenders and arrested prisoners and generate a DNA database like CODIS in the US. During the investigation, all crime scene collected DNA samples could be used to align with the profile in the database to discover any possible suspect. Ethics strongly underpins forensic analysis, specifically regarding 1. The use of DNA databases, 2. Privacy, and 3. Contamination. National DNA databases like CODIS have raised controversy surrounding its use due to the possibility of human subjectivity and error and biases, which lead innocent people and families to be linked to crimes they did not commit. This is exacerbated by the growing field of ancestry, which leads to continual DNA being added into third-party DNA databases, which upon a court warrant being received can be accessed to assist with forensic examination.
Privacy issues are raised where DNA from volunteers who help in identifying criminals also have their DNA added to DNA databases. This then raises the issue that the relatives of volunteers become traceable without their consent being obtained in the first place.
Finally, contamination of DNA. Where foreign DNA mixes with case-related DNA can in many cases lead to the wrongful conviction of an individual. Although the incidence rate of crime scene sample contamination is only about 0.75%, cases of DNA contamination can cause public uproar and reduce the validity and trust in forensic evidence presented to the courts in future trials. For instance, in Melbourne 2005, Gemma spent 18 months in the jail because her DNA sample was taken and placed at the same time as crime victim.